So in this webinar, I will talk about the combined influence of exercise and diet on the gastrointestinal microbiota. We know approximately 100 trillion microorganisms, most of them are bacteria, exist in the human gastrointestinal tract. The gut microbiome is now best thought of as a virtual organ of the body because it encodes millions of genes, producing thousands of metabolites. These metabolites replace many of the functions of the host, influencing the host's physiology and health. So what does intestinal bacteria do? Essentially, the gut microbiota allows fermentation of non-digestible substrates like dietary fibers, intestinal mucus, and this fermentation supports the growth of microbes that produce short-chain fatty acids and gases. Although we know that there is a heritable component to gut microbiota, environmental factors like mode of delivery, drugs, diet, metabolites, host immune function are also known determinants of microbiota composition. So each of us has a gut microbiome that looks slightly different to our parents, siblings, or friends. Our microbiome essentially is like a bacterial fingerprint specific to an individual. In humans, studies have shown the associations between microbes and health predictors. But we still need to do animal studies because they can help identify the mechanisms behind the link between gut microbiota and health, though the degree to which these findings translate to humans is still unknown. So what is the gut microbiome composed of? The gut microbiota consists of bacteria, archaea, fungi, protozoa, and viruses. Based on sequencing techniques, the dominant bacterial species in the human gastrointestinal tract are divided into three phyla: the phylum Actinobacteria, Bacteroides, and phylum Firmicutes. Other bacteria such as Lactobacilli, Streptococci, and Enterobacteria are also found in small numbers. Now, let's talk about the role of gut microbiota in health and disease. In some instances, the gut microbiome becomes imbalanced or disrupted, and this is what we call dysbiosis. Dysbiosis puts the host at risk. So it compromises the barrier lining the gut, triggering the immune system, activating inflammation, putting the host at risk for a range of diseases such as metabolic syndrome, obesity, inflammatory bowel diseases, liver disease, infectious diseases, diabetes, atherosclerosis. Lower bacterial diversity has been observed in individuals with inflammatory bowel disease, with atopic eczema, celiac disease, obesity, and diabetes compared to healthy controls. And this suggests that the greater the microbiome richness, the more robust it is against environmental influences. And the reason for this is that functionally related microbes can compensate for the function of other missing species. But the clearest evidence of the functional role of the gut microbiome in humans has been shown using fecal microbiota transplantation. So fecal transplantation is now the recommended treatment option for patients with recurrent or refractory Clostridium difficile infection. And this has been routinely used now in many hospitals worldwide. This is a schematic representation of the role of the gut microbiota in health and disease, giving some examples of the inputs and outputs. For example, intake of dietary fiber, probiotics can increase short-chain fatty acid production, improve lipid metabolism, decrease gut inflammation, reduce risk of infections, and decrease gut inflammation. In contrast, altered pH, excessive protein consumption, sugar intake, saturated fatty acid intake, and some medications can cause the opposite effects. I mentioned that diet is a determinant of gut microbiota composition. How does diet influence the gut microbiome? The type and amount of protein, fat, carbohydrates present in the diet have been widely found to impact the composition of the gut microbiota. Short-chain fatty acids, acetate, butyrate are reported as the end products of microbial degradation of protein and carbohydrates in the gastrointestinal tract. In fact, short-chain fatty acids produced by gut microbiota are the most extensively studied metabolites of diets and have been found to have an effect on the health of the host. This table summarizes some of the studies showing the influence of diet on gut microbiota and on host health. For example, high protein diet increases bacteroides, decreases firmicutes, causing decrease in body weight. Western diet, so this would be composed of high fat, high carbohydrate diet, causes the opposite effects. Basically, diet affects the host health by modulating the composition of the gut microbiota. These changes in gut microbiota then contribute to host physiology by producing various metabolites. These metabolites then exert their effects within the host and contribute to disease development. These are the methodologies commonly used to study the gut microbiome. So fecal or gastrointestinal content samples can be used for metabolite analysis, or they can be used to extract DNA, nucleic acids, which can then be used for targeted genomics, shotgun sequencing, 16S ribosomal RNA gene amplification. We already talked about gut microbiota in health, disease, and diet. Let's now discuss the influence of exercise on gut microbiota. Research from our group and others suggests that exercise also influences the gut microbiota. In fact, several animal studies have shown that exercise independently alters the composition and functional capacity of the gut microbiota. Matsumo et al. Was the first to find that 5 weeks of exercise resulted in an increase in the metabolite butyrate. And butyrate has been shown to increase epithelial cell proliferation, promote gut barrier integrity, and regulate host immune system and gene expression. In humans, evidence for the role of exercise in gut microbiota first came from cross-sectional studies. Clark et al. Found that gut microbiota of professional rugby players actually had greater alpha diversity and higher relative abundance of different bacterial taxa than the gut microbiota of lean, sedentary controls. Longitudinal studies, on the other hand, suggest that exercise has independent effects on the gut microbiota, but longer duration or higher intensity aerobic training is required to induce significant changes. Although several studies have shown that exercise alters gut microbiota composition, functional capacity, and metabolites, the effects of different exercise frequencies, different exercise modes, intensities are still unknown. Information on the effects of exercise on the gut microbiota in different populations and its effect with different diets are also lacking. Mechanistic studies and whether exercise induced changes in the gut microbiota are potentially disease modifying are also areas to be explored. This is a proposed model on how exercise alters the gut microbiota. So exercise has been shown to increase butyrate producing bacteria, increase butyrate concentrations, reducing pro-inflammatory cytokines, oxidative stress in the intestines or in the gut. So exercise is known to have protective effects against colon cancer, inflammatory bowel disease, depression, anxiety, obesity. But whether this disease protection is mediated by exercise-induced changes in the gut microbiome remains to be determined. Let me first define the different categories of exercise intensity. These categories are ranked according to energy demands using these objective measures. Primarily, we use VO2 max or oxygen, maximum oxygen uptake. So VO2 max is a measure of the maximum amount of oxygen your body can utilize during exercise. METs is another frequently used objective measure. MET stands for metabolic equivalent. This is an estimate of how many calories are burned during physical activity. So sedentary activities would be defined as less than 20% VO2 max, which would involve sitting or lying or activities with limited movements or low energy requirement. Light intensity activities would have 20 to less than 40% VO2 max. This would include domestic or occupational tasks. For example, washing dishes, hanging, washing, ironing, cooking, eating, working at a computer desk. So these are examples of light intensity activities. Moderate intensity activities, 40 to less than 60% VO2 max. Examples would be walking for a duration of 10 minutes or greater, gentle swimming, social tennis, golf, carrying light loads, bicycling. So these are examples of moderate intensity activities. Vigorous intensity activities, 60 to less than 85% VO2 max, are examples would be jogging, cycling, aerobics, competitive tennis, heavy lifting, aerobics, or fast bicycling. And then finally, high intensity activities would be exercise with 85% or more VO2 max. For example, running at a speed of 17.5 kilometers per hour. So let's face it, technology advancement in the modern society has caused large proportions of the population to adopt sedentary behavior. So a lot of us are sitting most of the time. So in fact, in a recent study, it has been shown that most people would sit 8 hours to 11 hours per day. So there's a growing interest in the health benefits of different activity intensities to promote public health. And two approaches currently very popular being investigated are high intensity activity and light intensity activity. So why is there growing interest in these two forms of exercise? Many people often find it more attractive or more attainable to perform light intensity activities than moderate intensity activities. HIIT, on the other hand, compared to moderate to vigorous intensity physical activity, this would require less time. For example, the Tabata workouts would only take about 20 minutes to complete. So that's why these two are quite popular in compared to moderate to vigorous physical activity. So just a quick survey. How much physical activity do you think is recommended in adults to achieve health benefits? One. Okay, so can you see the results of the poll of the survey?
Yes.
Okay, so it says here that about 40% answered A, which is 100 minutes per week of moderate intensity activity, and about 45%. So at least slightly more than 45% answered B, which is the correct answer is at least 150 minutes per week of moderate intensity activity or 75 minutes per week of vigorous activity. So either B or D is correct, or a combination of both spread throughout the week would be fine, would give health benefits. So the current literature so far have not compared the effect of different activity types or different intensities and diets on gut microbiota, which is important in understanding how changes to activity and diet can influence gut microbiota and in turn disease risk. Hence, to shed light to these questions, we investigated the intestinal microbiota responses of rats fed with high-fat, high-fructose diet. So this is the so-called Western diet, and the normal diet, standard chow diet, for a duration of 12 weeks using 4 activity conditions. So controlled, sedentary, high-intensity interval training, and light-intensity training. Sedentary rats were not exercised and were housed in small cages to limit physical activity. Light-intensity trained rats were trained at 8 meters per minute for 125 minutes divided into 4 bouts separated by 2 hours of rest between bouts. And this is performed 5 days a week. HIIT-trained rats were performing 4 2.5-minute work bouts at 50 meters per minute with 10% incline, each separated by a 3-minute rest period, again 5 days a week. And then we analyzed the microbial communities using KIME. OTUs or operational taxonomic units were clustered at 97%. Similarity using UCLUS. Pintail was used to inspect for chimeric sequences, and taxonomic assignments were used against as taxon. Statistical comparisons between groups were done using KIME-based ANOVA. So just to give you an idea on how light-trained rats were performed. I don't think I can play. I don't think I can play the video. Anyway, there. So that's how light intensity trained rats were exercising. And then just to give you an idea of the high intensity training, this is how fast the rats were training on the treadmill. So results of the study show that differences in community richness were not significant at an OTU or at genus level. OTU, or operational taxonomic unit, refers to clusters of organisms grouped by DNA sequence similarity. But richness index was significantly different at a family level between diet treatment groups. So this is the high-fat, high-carbohydrate diets are colored red, and the standard chow diet samples colored blue. So rats fed with different diets were significantly different at the phylum level, while there were no significant differences at the phylum level between exercise levels. So we saw significant differences if we compare different diets, but not between exercise levels. This figure shows hierarchy relative to abundance in standard chow and high-fat, high-carbohydrate diet. So the thickness of the branch is proportional to relative abundance. As you can see, firmicutes are more abundant or thicker in standard chow than in high-fat, high-carbohydrate fed rats. And then tenericutes would be more abundant in the high-fat, high-carbohydrate diet than the standard chow fed rats. These are PCA plots to observe the overall relationship between activity groups. As you can see, there is an overlap between exercise rats. So lit-trained rats, HIT rats would show overlapping microbial communities. So, and there will also an overlap between control and sedentary group. All communities from rats fed with high-fat, high-carbohydrate diet are overlapping, regardless of activity intervention. Meaning, rats fed with high-fat, high-carbohydrate diet have very similar communities, microbial communities, regardless of activity intervention, regardless of the exercise they are performing. Whereas standard chow diet allowed differentiation of microbial communities according to activity intervention. The HIT fed with standard chow have the highest similarity in microbiota between rats within the group, but distinct to other activity groups. Other activity groups, sedentary, light intensity trained rats, induce greater differences between rats within the group. This figure is showing differentially abundant families with stronger preference for standard chow diet. And as you can see, in standard chow diet, the abundant families were Lacno, Spiraceae, Rumino, Coccaceae, and Turrisibacteriaceae. While in high-fat, high-carbohydrate diet, differentially abundant families showing strongest preference were Erysipelotrichaceae, Corynebacteriaceae, and Bifidobacteriaceae. Now, let's look at the responses of activity groups in standard chow diet in more detail. There were no differences in alpha diversity between activity groups in the standard chow diet. So when we talk about alpha diversity, this is a measure of the microbiome diversity applicable to a single sample. However, in standard chow diet, beta diversity difference between standard chow groups was very high. There were more differences in OTU abundance rather than the presence or absence of different phylotypes between activity groups. The phyla, Tenericutes, Actinobacteria, Firmicutes were different between activity groups. Particularly, Tenericutes and Actinobacteria were induced in light intensity group with no significant change in high intensity interval training. So the OTUs significantly affected by exercise level in rats fed standard chow diet is shown in this figure. The closest isolates that increased in standard chow fed rats include Parasuterella excrementi hominis, Lactobacillus johnsoni, which is both increased in light intensity training, and then Clostridium saccharolyticum, increased in high intensity interval training. Now, in regards to responses of activity groups in the high-fat, high-carbohydrate diet, there were no differences in alpha diversity between activity groups, but there were significant beta diversity differences between groups. And the greater differences were found in the presence, absence of phylotypes rather than the change in abundance. So this is the difference between the standard chow diet and the high-fat, high-carbohydrate diet. In the standard chow diet, you have significant changes in the abundance, while in high-fat, high-carbohydrate diet, instead of the change in abundance, you have greater differences in the presence or absence of phylotypes. And the meaning of this indicates that the different activity intervention in the high-fat, high-carbohydrate communities had different participation in terms of bacteria. In terms of isolates, there were a number of species of Clostridium induced by light intensity training and high intensity interval training, including members of the family Clostridiaceae. So to summarize, our results suggest that light intensity training and high intensity interval training can induce microbial cohorts that break down carbohydrates more effectively. So the microbes or microorganisms increased in lit and heat are those that increase the breakdown of complex polysaccharides or complex carbohydrates, potentially improving the performance of energy provisions to working muscles. And the isolates that were increased in this exercising rats were also increased production of short-chain fatty acids. And this is good for the gut health, increased production of short-chain fatty acids. Now, the high-fat, high-carbohydrate diet induced similar but much weaker changes to microbiota, demonstrating the superior influence of diet over activity on microbiota response. So high fat, high carbohydrate diet negatively influences the benefits of exercise that may come from reshaping the intestinal microbiota, allowing only changes in the presence, absence of phenotypes in response to exercise. Collectively, these findings show that physical activity and diet impact on intestinal microbiota. However, their relative contributions to health and disease is still unknown. At present, increasing evidence suggests that regular exercise confers benefits to the gut microbiota. However, as you've noticed, there are still many questions that are yet to be answered. Particularly, what frequency, what mode or intensity of exercise would be best? How does exercise impact the gut microbiome in healthy or diseased individuals? What are the effects of exercise on gut microbiota in various regions of the gastrointestinal tract? How does exercise interact with diet in shaping the gut microbiome? What are the mechanisms behind these associations? Future research should also seek to understand which metabolites produced by the gut microbiota might be most affected by exercise training. Importantly, animal studies should also be explored to establish how exercise-induced alterations in the gut microbiota are linked to disease risk. Ultimately, we hope to see baseline gut microbiota, diet, and other host factors helping predict which exercise might be most effective for a given individual. So that ends the webinar. Thank you.